All right, biomedical innovation class. Mr. McFall going to go through e-learning for February 9th. The primary thing that you need to focus on for today is your Unit 6 lab report submission. So getting that turned in. One question we had yesterday about the template was the results section and what should be included in that. So I wanted to go over that real quickly. Let me open this in a new tab. So I think the summary was pretty clear. As far as results go, you're not going to have a lot of results to talk about in Module 2, 3, or 4, right? The main results are Module 1, talking about the gel analysis, prove that ligation happened, and then Module 5, talking about what these results specifically mean. And remember, in the results section, you're not really talking about like interpreting the results. You're essentially stating what the results, like how to, essentially what they are. You're objectively stating. So Module 1, a gel image, which you should have in your lab notebook, or you should have recorded. I will also insert an image of that into Canvas. I'll title it Module 1 results. I'll add that in there right after this so you guys can have it. And so you can see what that gel looked like and then be able to interpret it by looking back at the other parts of the lab. Remember, basically, I'll just go ahead and bring that up real quick while I'm on here. When you the results for that should show that ligation was successful. So if you look at this gel, this is the one from Module 1. We had our standard marker, but then this is the ligation control. We did not add ligase here. We had our plasmid, which is 3000, and we had our kanamycin gene, which was 1300. And so that's why you see two bands here, and then we see one large band proving that ligation was successful. All right. And then the Module 5 results, we went through in pretty strong detail. So essentially, you don't necessarily need this image if you can or you could draw the image, or you could take a screenshot of this if you want to, if you feel like you need to talk about it. So you should be able to handle that, I think. So that's really all you talk about in this Module 2. Basically, look at your results section of your organizer for the lab, and you should be able to explain, like, okay, Module 2 is basically one sentence of what happened. Module 3, did it grow or not? Module 4, did it appear to work or not? So there's really nothing much to write in Module 2, 3, or 4. 1 and 5, you should have more details about what the gels show. And then the under results. And then in the analysis, that's where you go into more details about what everything means. So once again, turn that in today. That's still due today. So you should be able to get that turned in to Canvas by the end of the day today. And then the next thing I want you to look at, just begin looking at, because we're getting close to the end of the trimester, is the statistical analysis of 213. And so there's making results meaningful, which is the name of the lesson courses. So I made this assignment, and I put some links into courses here. They should work. If you log into PLTW, then these links should take you there as long as you're logged in. So there's two tabs you need to have open in addition to the assignments. So what I would look at first is this statistical analysis resource sheet, which gives you lots of kind of background on terminology. And so it talks about what's called a null versus an alternative hypothesis. And really, when you start an experiment, you have a null hypothesis where basically comparing group 1 and group 2, if there's no difference and there's no effect of the independent variable, then those numbers are going to basically be the same. The alternative hypothesis is basically saying that alternative hypothesis is basically saying one is going to be better than the other, or that they're not going to be equal. So an example is like pharmaceutical says that it's going to enhance growth in children with human growth hormone deficiency. So you have two groups of children. One group gets the experimental, one gets a placebo. So the variables, you have the mean height of the experimental group and of the placebo group. Null hypothesis says there's no difference. Alternative says group 1 will be taller than group 2. So you're making a prediction, right? And so basically, you're trying to you're not trying to prove the alternative hypothesis, you're trying to reject the null hypothesis with your data. And if you fail to reject the null hypothesis, then your alternative hypothesis is supported. So essentially, if we can show that there's a vast enough difference statistically between the heights of the individuals who got the placebo compared to those who got the experimental drug, then we can show that there's a statistical significance that the drug does work more effectively. And of course, um, the more participants in a study, the easier this is, and the greater the difference between the values of the heights in the placebo group versus the um, experimental group, and then the more significant it is. So this talks through the steps. I think these steps aren't as helpful until you look at an example, but use these here as a reference to help you determine if the results are statistically significant or not. And this is basically what's called a t-test. And so there's two other things here. This is the 213 examples. If you click on this goes through 4 different examples. We're going to do example 1 as just kind of because it's given on the assignment. And then there's another one later that we'll do next week. And so the other one that we'll do next week relates a little bit more to your test, because you have to pick, there's different types of t-tests that can be done. And if I go back to these notes, the type of t-test that would be done could either be independent t-test, where you have 2 different groups being tested for the same variable, like a placebo versus experimental group. And knowing your experiments, you did not do that, and that's fine. Your experiments involved using the same people testing the same person before and after, like a pre and a post test, okay? And so that's called a paired t-test or dependent t-test. And so essentially, we're going to be using this equation where we're using the same participants in the end. Don't get intimidated by this. We'll break this down so you can do this and know what it means. Also, this can be done in Excel or Google Sheets, but we're going to go through the hard work to understand why it works. Anyway, so this is the experiment that you'll be doing, and if you look at the examples, I think it's example 3 that has a dependent, this has a dependent t-test. So where they had individuals who had to wait before treatment and after treatment, so you're testing the same person. So we'll go through example 3 next week together, since that one's more like what your experiment was. But let's go back, and you're going to go through example 1. And on the paper I gave you yesterday, if you brought it home, if not, here's a document. If you don't have it, you don't necessarily write it down today. You can also catch up on this later, but I would encourage you to at least try to write down the steps of the t-test statistics assignment. You'll go through the steps, which I have it open over here. And here's example 1. It talks about if males are more likely than females to develop a cold. And so there's 30 subjects. This would be like a prospective case study, and to determine how many colds they develop. And so you can see the data for males versus females and all the steps. And basically, you go through the null and alternative hypothesis. So if you look in the example, so example 1 is exactly the same thing, and it basically talks you through each of them. Well, the null hypothesis is females develop the same number. Alternative is males develop fewer colds than females. All right, and so that you would fill in this information as you go. Step 2, the level of significance. Well, in this case, the alpha value is always going to be given for you. And for our purposes, we're going to use 0.05. That's important for whenever you're determining what's called the t-statistic. And then you calculate the mean, variance, and standard deviation, all some great math terms I'm sure you love. And I'll show you the Excel example you can give. You don't need to write out all of this work. just like the main numbers. See what's in bold. Those are kind of like the final answers that are important for this. We'll go through this a little bit more detail. So just read through as best you can, and then fill those out as you can see on the document. Step 3, you'll record the sum of the samples and calculate the means. You'll calculate the variances and the standard deviations, and just record those. Step 4, determine which type of two sample t-test is most appropriate. Well, it tells you in here that it is an independent t-test. Okay, and so this would be the one that's most appropriate. And then you calculate this t-statistic in degrees of freedom. And a lot of this is probably brand new stuff that maybe isn't the easiest to get right now. Then step 6, use the t-table and t-statistic to get the critical values to determine whether you reject or fail to reject the null hypothesis. So from there, basically state whether they are or not significant. And with that, if you look at this, for example, you see that you do the calculation, the degrees of freedom ends up being 28. The t-statistic is 0.484, and our alpha value is 0.05. So we're always going to use this column, 0.05 for the alpha value. And our degrees of freedom is 28. Well, we see this number 1.701. So because the value of the t-statistic is 0.484, this number, it's less than the critical value. So we fail to reject the null hypothesis, meaning this number is not high enough. This number has to be higher than 1.701. That number is higher than 1.701. Ultimately, that means that there's a big enough difference between group 1 and group 2 to say that the results are statistically significant. So based on our data, our results are not statistically significant between the number of colds between males and females. I'll go through that a lot more. That may not make much sense right now, but if you can just try to fill out through the part 1, and we'll go through part 2 in detail together in class next week sometime, probably next week, as you interpret those results, depending on when we get back. But nothing turning on this right now. The main thing is turning in your lab report. So get that done and turn that lab report into Canvas, and that is it. So email me if you have any questions, and hopefully I'll see you soon. Thank you.